Welcome everyone, and thank you for joining us today for our webinar titled Automated Quantitative Measures of Forelimb Function in Rats and Mice. This is Andy Henton from Inside Scientific, and I will be your host for today's event. Our session is sponsored by Vulintus and will focus on training and testing protocols of forelimb assessment in rodents using the Vulintus MotoTrack behavioral system. First, we will be joined by Dr. Drew Sloan, Chief Operating Officer and one of the founders of Vulintus Incorporated. Drew completed his PhD in bioengineering and following conducted extensive research focused on trained rat motor behavior. As an engineer, naturally he wanted to make behavior training and testing as efficient as possible, so along with many research collaborators, he helped to develop some of the automated forelimb assessments you're going to learn about today. Following, we will hear from Dr. Seth Hays, Assistant Professor of Bioengineering at the University of Texas at Dallas. His current research is focused on developing vagus nerve stimulation therapy to improve recovery of motor function after stroke, traumatic brain injury, peripheral nerve injury, and spinal cord injury. Today, Seth will share some of his research findings related to the use of the MotoTrack system and discuss how these findings are translating to clinical application.
So we're going to be talking about automated forelimb assessments in rats and mice today. One quick caveat, my research background is almost exclusively with rats, and a lot of the tasks we're going to talk about today were first developed for rats, so you're probably going to see a lot more rat data than mouse data. However, we do have mouse systems that are in use by several labs. Publications are just starting to come out about that. Excuse me. So if you have any questions about rats versus mice in particular and our mouse systems, if you stick around to the Q&A at the end of the talk, I'd love to talk to you about those if you don't see enough mouse data here in the talk. So let me give you a quick preview about what I'm going to talk about. We're first just going to very briefly talk about automated forelimb assessments in general. After that, I'm going to give you a very brief overview of Vulintus' MotoTrack system that we use to run the behaviors I'll be talking about. And then the bulk of my presentation is going to be kind of like a methods walkthrough of how the behaviors are trained and tested. We're going to go over three different tasks: the isometric pull task, the supination task, and a lever press task. I'm going to mostly go in depth about the isometric pull task because that has the biggest publication history. It's been used in the most models. And then for the other two tasks, I'll mainly highlight the differences in how those tasks are run relative to the isometric pull task because they have a lot of commonalities. And then after that, I'll turn it over to Dr. Hays, who's going to tell you about how he's used some of these assessments in his research to translate his preclinical work into clinical studies. So first, let's look at non-automated forelimb assessments, and these should be familiar to most of you. First of all, I should apologize because I'm positive that I kind of left a bunch out of here. I kind of try to generalize. The top three tasks, pellet retrieval, staircase reaching, and pasta matrix and handling, are all variations on trained reach and grasp and retrieve motions. Cylinder and ladder rung, or rope walking, or treadmill walking, like somebody said in the comment window, they assess forelimb placing and forelimb placement accuracy. And then you have what you might call semi-automated tasks such as grip strength, where there's still a lot of experimenter hands-on to it, but you get quantitative measures. Now, one common element across all these tasks is that they require focused experimenter supervision, and it's often one-on-one, one experimenter to one animal. That's either to set up and run the test or to score the test, often from looking at video afterwards, or sometimes it's both. And the one-on-one ratio of animal to experimenter, that requires a lot of manpower. So you either have to have lots of graduate students or undergrads to run it, or you're just limited in the amount and the frequency of data collection that you can do. And also because you're doing a lot of manual scoring, data is mostly qualitative, which can increase the variability when you've got multiple researchers that are handling groups of animals within the same experiment. So within the last several years, several groups have tried to create and have reported how they've designed various automated forelimb assessments to try and solve some of the problems with these more manual tests. Now, again, I've probably forgotten a lot of groups here, and I apologize. This is just a very brief survey. I've roughly categorized system based on task goals. You have some tasks that try to automate common manual forelimb assessments directly, such as pellet retrieval. And on the other end of things, you have tasks which train animals to manipulate relatively complex interfaces, such as joysticks. And in the middle, you have the three tasks that I'm going to be talking about today. So not to belabor the point too much, but comparing common, more manual forelimb assessments to automated assessments, common forelimb assessments do have advantages. There's a very low equipment cost, so just about any lab can start them up. All you need is a specialized cage and the manpower to run the task. And you can do relatively complex parameterization because manually scoring can be broken up to score very subtle differences in movement. So you can identify things that machine vision or various sensors couldn't identify. There are limitations though, like I talked about, chief of which is that they're supervision intensive. This in turn can limit the number of subjects your lab might be able to test in a given day, so you have low subject throughput. And it also might limit the number of trials you're able to collect on every test, which can limit your statistical power. Most of these manual assessments require some kind of manual scoring, which can double or even more than double the supervision requirement, as you have to go back and basically re-watch the same behavior to score it. And this can also add variability between raters. When you have multiple people rating the same behavior, oftentimes you'll have to have the same behavior rated by 3 or more people just to come up with an average score. And there's also often a delay between when the behavior is run and a score. Oftentimes you don't have time to get to the scoring until several days later, and so you never really know exactly how good your behavior was until you can score it several days later. Automated assessments, by contrast, have several advantages. They're automatically scored, and you can get quantitative, objective measures of behavior. This reduces the need for supervision, and so one experimenter can run many subjects at once, so you don't need quite as much manpower to run as many animals. And with less supervision, you can get more trials with really no extra time cost, so you can get high counts, which can give you very high statistical power. And you can also get real-time analysis to show immediate feedback, so you can see that your behavior is working as you're doing the behavior. And with task presentation and scoring being automated, you need less experimenter training, so it's easier to have undergrads or graduate students kind of start running the behavior very quickly. And the automated scoring improves the replicability across similar conditions. So when you're running multiple experiments and you want to do the same behavior and compare them, it's a lot easier to do when your scoring is automated. It's not all upside with automated forelimb assessments, of course. There's a significantly higher setup cost. You need some specialized hardware. It's usually computerized. And to automate the behavior, you also need to parameterize it into a limited set of endpoint measures. There's going to be some aspect of the behavior you're measuring, and parameters that you're not looking at might be lost to you after the behavior is run. Whereas with videotape behavior, you can go back and reanalyzing it using a different scoring system if you think of that later. So, automated assessments have a lot of advantages, and maybe you're watching this webinar because you're thinking about using them in your research. There's a lot of options out there, and most of them have been described in methods papers, so you could build your own if you wanted to. I'm going to be talking about just 3 of these, however, the 3 that I've helped to develop, and I don't want to give you the impression that I developed these by myself, just sitting in the lab. I worked with a lot of people and a lot of collaborators, and I'm also giving this talk because I've had a hand in each of these, and so I can talk pretty easily about all 3 of them. And all of them have been reported in methods papers that you can also download and take a look at. So the tasks that I'll talk about were first developed in the lab, primarily at UT Dallas, but are now being developed by Vulintus. And we spent the last couple of years trying to create a unified system that can run all of these assessments, and we've got even more assessments in development. And the system that we've combined these all into is called MotoTrack, and it's a modular system so that it has a lot of common components, and the way that you run the different tasks is by swapping in different task modules. To give you a little bit of history about the MotoTrack system, again, it was originally developed at UT while I was doing my postdoctoral work there. We created these automated tasks, and we had a lot of interest from other labs that wanted to do the same thing, and they really wanted to be able to get an off-the-shelf system. So we created a company to see if we could mass produce the system at a relatively low cost. NINDS thought this was a pretty good idea as well, and they gave us a small business grant to help develop MotoTrack. We originally developed MotoTrack with rats in mind, but in the last 2 years we've really expanded the system so it now works with mice. And to date there's about 16 research papers and counting that have used MotoTrack in a variety of forelimb impairment models, from ischemic and hemorrhagic stroke, traumatic brain injury, spinal cord injury, and then more basic neuroscience like motor learning. And there are several models on the way, including peripheral nerve injury and probably several I'm not even remembering. It's being used in quite a few labs around the country and even in Europe. So in the development work that we put into MotoTrack in the last several years, we focused on integrating several key features. And the first key feature we've emphasized is interchangeability. One MotoTrack system can run all the tasks that I'm going to talk about. And different task modules can be swapped in and out to run the various different behaviors on the same base cage. And likewise, that base cage can be quickly configured for either left or right paw or either paw configurations. So you can really set it up to run the behavior exactly how you would like it to be run. The second key feature we've emphasized is consistency. I don't want to badmouth undergraduates or graduate students, but when you've got a lot of different people working on one study, a lot of variability can creep in just by how different people handle things. So to minimize that, we've tried to automate as much as possible, from the physical manipulative positioning to file organization, so that all the system really needs to know is what animal is going to be tested and what test should be run on that animal, and then all other aspects of the behavior should be automated. You really just need to put the rat in, take the rat out, and enter in the name. And the third feature we've tried to emphasize is real-time analysis. You should see the measurements coming from your subjects as they're being collected so that you can verify that the behavior is working and you can get a sense for how your animal is performing during the behavior itself. Like I said, MotoTrack has already been used in 16 publications and counting. It's been used across many models, including stroke, traumatic brain injury, spinal cord injury, and others. And in all these tasks, using both the isometric pull task, the supination task, and the lever press task, they've been able to detect significant lasting impairments lasting out to several weeks. But to show that the automated tests are testing much of the same function as the common tasks, we've also cross-validated the isometric pull task and the lever press task against skilled reaching, plasticity, and ladder rung walking in the same animals. And what we find is that the deficits are correlated. Animals that have a deficit on the common, more manual tasks have a deficit on MotoTrack as well. However, the common, more manual tasks tend not to show the same lasting impairments that are detected by the automated tasks. As we look out to several weeks following injury, the variability in the more manual tasks tends to mask the significance of the impairment, whereas with the automated tasks, the low variability lets you see the significance of those lasting impairments. So let's talk a little bit more in depth about how these tasks are run. And the first task I'm going to talk about today is the isometric pull task. And I'm going to go into some depth about this one because this is the task that's been used in the most models and has the most publications. After we talk about this task, then we're going to talk about the supination task and the lever press task. We're going to talk about how the methods differ for those tasks from the isometric pull task. The walkthrough will be similar to how a new user might approach the isometric pull task. We're going to first talk about the system setup and what the overall objective of the task is. Then we're going to talk about training considerations and what we consider to be criterion level performance, where you're ready to test your particular model. And then finally, we're going to talk about considerations for post-impairment testing and analysis of the data. For the isometric pull task, the setup is that the rat reaches through a slot to grasp a stationary handle. That handle is connected to a precision force sensor, which measures the force applied to the handle. And when a specific force threshold is exceeded, the system can trigger a food reward pellet. The animal will go to get the pellet and will then come back and reinitiate the behavior. And they can do this several hundred times a day. Graphically, when we look at the signal coming from the force sensor, this is what the behavior looks like. Animals tend to go from grasping to pulling very quickly. In healthy animals, a specified force threshold equal to about 40% of the body weight, which in rats is about 120 grams and mice is about 20 grams, that can be applied very quickly. Animals will often pull with that amount of force in about half a second or less. The force profile can look different from trial to trial, oftentimes with multiple attempts before the force threshold is exceeded. I should also say here too that you're not limited to just a hit criteria of exceeding a force. Because we can analyze this signal in various different ways, you can also create behavior where animals have to reach and hold for a sustained pull, or they have to generate multiple peaks, and you can really dial in that sort of behavior however you'd like it, however we can analyze that signal for you. Of course, a video is usually the best way to show behavior. So here, we're going to see a trained rat doing the isometric pull behavior. This is courtesy of the Renneker lab here at UT Dallas. You can see the rat reaching through the slot, grasping the handle, and pulling. And then they go over to get the food pellet. You notice that we use a partition to force right paw behavior for this particular animal, and the partition also forces the animal to go around to get the food pellet, and that causes them to reset their stance when they come back to do another reach. Like I said, the isometric pull task has also been adapted for mouse models. And a couple of modifications had to be made to the setup to make it work. First of all, we've adjusted the task dimensions, so the floor of the cage is raised up with a removable riser, which brings the manipulandum to a more accessible height for the mouse. A smaller handle is also used to better fit the mouse paw, and we've used a more sensitive force sensor to detect the smaller force range of the mouse. Additionally, some of the labs we work with prefer to use liquid rewards for mice. Mice satiate very quickly when you use those standard 20 milligram food pellets, and you can often only get maybe 50 trials per day. With liquid rewards, you can aliquot those out to motivate trials in the range of hundreds per day, and so a lot of labs tend to like to use liquid rewards. Again, a video is the best demonstration, so for comparison to the rat, here's a mouse doing the isometric pole behavior. This video comes courtesy of April Becker from the Goldberg Lab at UT Southwestern Medical Center. April just recently published a methods paper on this mouse behavior that you might have read. And so in the video here, you can see the mouse reaching and grasping the handle and pulling. And you can see that the mouse doesn't necessarily get a full grip with all the digits. Typically, a grasp involves just several of the digits. So, now that you've seen what the trained behavior looks like, you might be interested in the steps you need to go through to get an animal to the point where they're doing that behavior. Training is organized into stages, with animals run on one stage per session, and then as they graduate each stage, they get started on a new stage in subsequent sessions. So with progressing stages, various parameters can be adjusted to train the animals to first interact with the pull handle, and then to pull with sufficient force. And then finally, you want to make sure that the animal is reaching far outside of the cage to interact with the handle so that they get a full reach. Additionally, we can adjust the reward schedule to increase or decrease feeding frequency to maintain motivation. And we can add programmable auditory cues which provide feedback for the animal during training, and you can shut those off during testing if you're worried about any kind of auditory artifact. Parameters also don't have to be static during a session either. Typically, we adaptively vary the force threshold to allow fast or slow learning animals to progress at an individual pace, because I'm sure you've seen that individual animals definitely have differences in how fast they can learn. Sometimes you have smart, fast learning animals and sometimes slower learning animals. Before training can really begin, some initial shaping is required to create the association between the pull handle and the reward. We have tried to automate this as much as possible. So if you're a patient, you can usually accomplish this with just one or two somewhat longer, about an hour-long shaping sessions, which you put the animal in the cage, and they're going to learn the association between the handle and food pellets with just incidental encounters. When they bump into the handle, it'll cause a food pellet to drop, and over enough time, provided that you're not watching too closely, you're not watching the pot boil, so to speak, you'll see that the animal forms the association. If you want to speed that association, though, pellets or other food can be used to kind of bait the handle, to kind of lure the rat towards the handle for those first interactions. And after that, it happens very quickly. So once training is in progress, once the animal's been shaped, again, the goal is you want to increase the amount of force applied to the handle by the animal, and you also want to increase the reach distance so that they're reaching farther out. Most of the labs we work with do 2 30-minute sessions per day, a morning and afternoon session, with at least about 2 hours between session. Most labs do behavior about 5 days a week with free feed on the weekends, although some do 7 days per week if you're really if you're really into working on the weekends. And during training, we found it particularly effective to adaptively vary the force threshold during the sessions to maintain about a 50% reward schedule. That maintains vigilance. But we also apply a ceiling to the adaptation to emphasize control somewhat over brute strength. You don't want to have an infinitely increasing force threshold because you're going to turn these into little Arnold Schwarzenegger bodybuilder rats if you do that. And for the isometric pull task, it takes about 15 days to train a naive rat from initial shaping up to the point where they're ready to do criterion level performance, and then about a week after that to establish a criterion baseline. Now, when we talk about criterion, I should say that there are different ways that you can run this test. It doesn't just be exceeding a force threshold. Again, you can do like a sustained pull, you can do a multi-pull situation. But just for example, probably the most common paradigm that's used with this task is that for a hit, a rat will have to exceed 120 grams of pull force within 2 seconds of initiation, and with mice that's set to 20 grams. They have to do this performance for 5 consecutive days at a level of 85% hit rate before we would consider them ready to do whatever sort of model you would want to test. And at that level of performance, they can do 250 to 300 trials per day. So you get a lot of statistical power just within one day. Following whatever impairment model you want to do, most labs will do home cage recovery of at least a week, more if you've got a particularly severe model or if you want to look at some sort of chronic impairment. For really severe impairment models, you might not be able to use the same pre-impairment performance criteria that after you've done the impairment. For example, this trace on the right is from a rat that was trained to pull with at least 120 grams of force, but following a cortical-subcortical stroke model, this rat can never pull with more than 80 grams of force. So in this case, we applied an adaptive threshold after impairment. That helped subject impairment, and it kept this rat working through 5 weeks of post-impairment testing. Whereas this rat was also trained on the manual skilled pellet retrieval, and this rat never completed a single successful retrieval after lesion, and they essentially stopped attempting that task after a week. Analysis can be done at any point and in an ongoing way. As soon as you have data from a single session, you can start doing your analysis. It's MATLAB based, and that's available either as script or a standalone program, so you don't have to have MATLAB to do it. It's open source. You're welcome to modify it in any way you want. We use graphical interfaces so that you're not just staring at numbers, that you can see the data in a graphical form. And the analysis is going to let you zoom right down to individual signal samples from individual trials, or you can zoom out to look at groups of animals over long time scales and kind of see how your interventions are doing. So that's the isometric pull task. That's basically the methods for that task and how we run it. Now we're going to talk about the supination task and talk about how that differs and how that's similar to the isometric pull task. The similarities start in just the setup. You can see it's the same base cage, and the main difference here is in the manipulandum and what exactly it is we're measuring. The manipulandum for the supination task is essentially just a rodent scale doorknob, and it's got some texture to it to allow a firm grip. Animals will reach through the slot, they're going to grasp the knob, and they're going to supinate in order to turn the knob to a specified turn angle, which we measure with a precision angle sensor. Now graphically, this is what the signal from the turn angle sensor looks like. And for the animal these traces were taken from, the supination threshold for what you consider a hit is 60 degrees. So they're reaching out, turning that knob for 60 degrees. And healthy trained rats can tend to complete the supination movement in one continuous motion. And it takes about a second from initiation to release. Sometimes it can take several attempts before they can complete that 60 degrees, however. And you can see from these individual trials on the right what some other types of trials that are not simple single movement might look like. Once again, a video is the best way to demonstrate the task. And so this video comes courtesy of Eric Myers from the Renneker Lab at UT Dallas. He just published a methods paper on this task just this last March, and you might have read it. And you can see the rat reaching through the slot, grasping the manipulandum, and turning 60 degrees to get a food pellet. And the rat can do this hundreds of times a day. So how this task differs from the isometric pull task when we're talking about training and testing, it's very similar in that it progresses through similar stages, but with some key differences. During shaping, the knob is left to free spin with very little resistance. We find that supinating against resistance is one of the major hurdles that an animal has to overcome during training. And so naive rats are started with very minimal resistance, just the resistance of the angle sensor itself. Once the rats are engaging the knob through successive training stages, they're required to turn the angle farther on every stage, and an increasing counterweight is added so that there's more torque required during supination. Training at this task takes approximately 1 to 2 weeks longer than with the isometric pull task. And once again, specialized performance criteria can be designed, but here we'll talk about just some example criterion where the animal has to just turn past the specific turn angle. You could also do, for example, like a turn and hold or a multi-turn sort of scenario. For example, though, you could train the rat to turn the knob through 60 degrees or more within 2 seconds of interacting with the knob. And if they can do that at over 75% performance over 5 consecutive days, we would consider them ready for whatever model you'd like to test. And similar to the isometric pull test, you can get 250 to 300 trials per day. Now these are the parameters for the rat, and I should note that we've recently started working with labs on a mouse version of this task, but this is very much in development. We're just trying to figure out exactly what sort of parameters will work with the mice and what sort of dimensions will work as well, and I hope to have some good data to show on that pretty soon. So finally, let's briefly touch on the lever press task, which is perhaps the most classically familiar task of the three. It's very similar in basic setup to the other tasks, and the manipulatum in this case is the lever arm. Animals will reach through the slot and depress the lever. And unlike other lever tasks, however, we measure the lever depression with a precision angle sensor instead of just an on-off switch. So we can detect all interaction with the lever in an analog fashion and not just full depressions. When you look at that signal coming from the angle sensor during behavior, you can see that the animal can very quickly achieve a full depression. And I should emphasize that, especially with these lever press tasks, because we're measuring press angle using the precision angle sensor, there's many parameters of the movement that you can use to create a behavior to isolate components of the movement. It doesn't just have to be some sort of multi-press paradigm. You can look at how smoothly the lever is pressed or how fast, and you can do these press and hold or sort of middle press range behaviors. Dr. Hays has used this task to quantify bradykinesia in several models, and the behavior shown here on these traces is a double press behavior in which a rat is trained to fully depress the lever twice in rapid succession. A trained healthy rat can easily complete 2 full depressions within about half a second. And some trials you can see from the individual traces here, they may include multiple incomplete depressions, or perhaps lingering movements. And these data points themselves are sometimes interesting from a data analysis standpoint, and they may be illuminating to your model. So, once again, a video is the best way to press the task, or a video is the best way to demonstrate the task. This video is courtesy of Dr. Hays himself, who published the original methods paper on this task and has been working with us through many versions of the system. So you're going to see some of his data from this task in his following presentation. You see the rat reaching through the slot and very quickly doing a double press. In a couple of trials, you might see him do more than 2 presses. He goes over to get a food pellet and then goes back and presses the lever again to initiate a new trial. So, looking at the differences in training and testing compared to the other 2 tasks, it's very similar again. During shaping, any small depression of the lever is rewarded. And during training, through successive stages, rats are trained to fully depress the lever. And then, if you want to do some sort of multi-press paradigm, they can be trained to press it multiple times in rapid succession. We can add programmable audible clicks to provide auditory feedback to the animal, and those audible clicks can be turned off in later testing if you're worried about auditory artifacts. Training is very quick. It typically takes 3 weeks. It can even be a little bit faster than the isometric pull task. And again, I want to emphasize that specialized performance criteria can be designed however you would like that task to be run using the signal coming from the precision angle sensor. But a typical paradigm might be where we'd consider an animal ready for lesion when they can fully depress the lever twice in rapid succession within about half a second, and when they can do that on 80% or more of trials for 5 consecutive days. And similar to the isometric pull task and the supination task, you get about 250 to 300 trials per day. And again, we also have a mouse version of the system, but it's out there in development right now, being tested by several labs, and I don't have any great data on this just yet. So, not to end too abruptly here for my section, but I've detailed some of the methods behind these automated tasks, and I'm going to turn it over to Dr. Hays here in a second to talk about how he's used these tasks in his research. At the end of the talk, I hope you'll stick around for some questions and answers, because I love to talk about these automated forelimb assessments. And I want to talk about how they can help you supercharge your research and really ramp up your experiments so that you can get a lot of subjects going, get some good statistical power and replicability. Vulintus MotoTrack system can run these automated assessments I've talked about, the isometric pull, the supination task, and the lever press task, and they all work within the same system. But we've got even more tasks in development, and if there's any particular motor behavior that you'd like to see automated, you should talk to me about it, and maybe it's something we're either working on or something we'd like to get into. So stick around for the question and answer session, and I'd love to hear your questions. Of course, if you don't want to ask me questions, you're feel free to contact anybody at Vulintus. We're still a small company, and we work closely with our customers to really make sure they're getting the most out of their systems. So if I don't have time to answer your question here, feel free to contact any one of us at any time.
Well, first of all, thanks to the organizers for giving me the invitation to speak today. I'm looking forward to sharing some exciting new research that my lab is doing that's regarding stroke recovery, and I want to touch on how it's been accelerated by these high-throughput MotoTrack systems. So specifically, my lab focuses on using cortical plasticity, and in particular ways in which we can manipulate or target plasticity to drive beneficial changes and hopefully improve recovery after neurological injuries or disease. So the brain obviously has a remarkable capacity to reorganize, but in the context of many neurological disorders, insufficient or maladaptive plasticity limits recovery of function. That being said, if we could find an adjunctive strategy to enhance neuroplasticity, we could potentially treat a lot of neurological diseases if we could apply this plasticity appropriately. So I'm going to focus my talk today on using targeted plasticity therapies to improve recovery after stroke. And stroke itself really needs no introduction. It's a leading cause of disability across the world, and in the United States it's one of the major causes. There's about 825,000 cases of stroke each year, and many people who suffer a stroke are left with significant disability. As a result, there's millions, it's been estimated at up to 4 million people in the United States who are living with stroke-related disability, oftentimes affecting motor function, which can lead to inability to perform activities of daily living. So the gold standard treatment that's given for stroke survivors is rehabilitative therapies. And so rehabilitative therapies are effective in some individuals, but they're not consistently effective across many individuals. So a lot of times this depends on how impaired you were at the beginning, immediately following your stroke, as to how much recovery you'll demonstrate. But it's certainly clear that given the amount of people who are left with disability, even after intensive rehabilitation, that we need to find better ways to improve recovery. So the way rehabilitative training is thought to work, or at least one of the ways in which rehabilitative training is thought to work, is that it drives adaptive plasticity or beneficial plasticity in motor circuits, and this plasticity is associated with recovery. So on the right-hand side here, you can see a schematized view, top-down view of the motor cortex, where in general some proportion of the neurons are devoted to controlling the hand, some to the head, and some to the leg. After an ischemic stroke event, a lot of times some of the neurons that are controlling these movements are damaged by this, and that leads to a loss of precise motor control. So with rehabilitative training, what happens is there's reorganization within the motor cortex that leads to now a greater representation of some of the impaired movements. And this is believed to, in part, confer partial improvement of motor control. So based on these concepts, it's possible that if we could enhance this reorganization, we might be able to boost the benefits of rehabilitation and increase recovery. So the concept behind this is, this is sort of a classical neuroscience idea where plasticity is it results synergistically from neuromodulatory release, so neuromodulators, things like acetylcholine, norepinephrine, dopamine, serotonin. The release of these neuromodulators in conjunction with neural activity leads to plasticity. So following up on this simplified idea, it's possible that if we could enhance neuromodulatory release, we might be able to enhance plasticity. And this is something people have tried to do for a long time. So here is a view of acetylcholine and norepinephrine, or two of the major centers in the brain that control release of acetylcholine and norepinephrine. So the locus coeruleus, located in the brainstem and shown in blue, controls release of is the primary source of norepinephrine in the brain, and the nucleus basalis, which is shown here in green, is the primary source of acetylcholine throughout the forebrain. And so through a variety of hard work by a lot of people that I sadly won't have time to cover today, my lab is interested in using activation of the vagus nerve to drive activity in these two neuromodulatory centers, subsequently releasing these neuromodulators and enhancing plasticity. So the concept here is that people have shown in the past that by stimulating the nucleus basalis or the locus coeruleus, you can enhance plasticity, but because these structures are located and they're so inaccessible, it would be really useful to have a non-invasive or at least less invasive way to activate these things. And so one of the things we've stumbled upon is that activating the vagus nerve drives activity in these nuclei. So we tested then whether we could use vagus nerve stimulation paired with motor training to enhance motor plasticity. And so the way we did that was we trained animals on a wheel spin task where the animals were charged to reach through a small slot in the cage and spin a wheel, and this primarily emphasized the use of the digits. And so again, here we're looking at a view of the motor cortex. and this is derived from intracortical microstimulation mapping. And you can see here that there are different sets of neurons that are predisposed to control either distal forelimb function, proximal forelimb function, head movement, or hind limb movement. And so this is what the organization looks like in a naive animal, just to give you some idea of what the general organization of the motor cortex. If we look at animals who receive vagus nerve stimulation paired with this wheel spinning test, what you'll see is that there's an increase in the area that's devoted to controlling the distal forelimb musculature. So we saw about a significant 30% increase in the amount of distal forelimb representation in the motor cortex. To show that this wasn't just sort of a generalized effect related to distal forelimb function, we trained a separate cohort of animals to perform another task. And this task is the lever press task, which Drew told you about a couple slides ago, where the animals had to reach and rapidly press a lever twice within 500 milliseconds to receive a food reward. And half of the animals who did this received VNS paired with successful trials. And when we assessed the reorganization of the motor cortex of these animals, what you can see here is that there was a dramatic increase in the amount of neurons that controlled proximal forelimb musculature. So this was about 159% increase on average. And this suggested to us that VNS not only could drive robust plasticity in the motor cortex, but that it was specific to the movement with which it was paired. So given this ability to drive this robust specific plasticity, we asked whether we could enhance rehabilitative training, whether we could promote plasticity to enhance rehabilitative training and subsequently improve recovery after stroke. So the concept is, again, based on this design where increasing neuromodulatory activity in conjunction with neural activity can enhance plasticity, but here we're replacing the neural activity with the neural activity that's driven by rehabilitation. And we're using VNS to enhance neuromodulator release, and we're testing whether we can use that to improve post-stroke recovery. So to do so, we designed the experiment in the following way. So you can see here, animals were shaped on the task. They underwent pre-lesion testing for 2 to 4 weeks, and then we measured their stable criterion performance, for this pre-lesion performance, for a week. Once they had reached asymptotic stable performance, we induced a stroke in the motor cortex using endothelin. This is a vasoconstricting peptide, which models ischemic stroke. So after we induced the stroke, we allowed the animals to recover in their home cage for a week, and then we brought them back for 2 days of post-lesion assessment. And based on their post-lesion assessment, they were assigned to balanced groups to receive either rehabilitative training over the course of 6 weeks, and the rehab consisted of, 2 daily sessions, 2 daily 30-minute sessions of freely performing the isometric pull task. The other group received rehabilitative training, the equivalent rehabilitative training, but they received VNS paired with successful trials over the first 5 weeks, and then on the 6th week they just performed rehabilitative training. And if we look at what the VNS pairing looks like here, you can see animals, here's a 30-minute snippet from a session. Animals who receive rehab wouldn't receive any VNS pairing during the session, but animals who receive VNS paired with rehab are going to get a short 500 millisecond burst of VNS paired with each successful trial. So let's take a look at the results. So prior to the lesion, both groups of rats were highly proficient at the task, performing over 85% correct. After the lesion, both groups were significantly impaired, demonstrating about a 50% reduction in hit rate. And if we look at performance over the course of 6 weeks of rehabilitative training, you'll see that animals who received this extensive rehabilitative training alone about a 50% improvement in recovery. So this is a modest but significant improvement, and this is kind of what we expected to see. So they're getting basically the gold standard in rehab, or the gold standard in therapies, which is extensive rehab, and we're seeing that many of them are demonstrating some improvements. If we look at animals who receive VNS paired with rehab, you can see that they demonstrated significantly greater recovery over the course of therapy when VNS was being paired. And when we look at performance on week 6, so at this point animals are no longer receiving VNS, you can see that these performance benefits are maintained, which is suggesting that this might be a long-lasting improvement in function. And so this was encouraging to us, but you need to be really cautious about things like this, because stroke therapy, or translation of therapies from preclinical stroke research to clinical stroke to use in stroke patients, has been fraught with challenges. And this is well recognized in the field that it's really difficult to translate these things. And one of the reasons it's thought to relate to this is that we need to do more work on the preclinical side, really vetting these therapies and really optimizing them, so when we do take them to the clinic, we're able to translate them effectively. And so I've combined some some of these guidelines that have come out, and there's a variety of different sources for these. There's the ARRIVE guidelines, there's the STARE, which is the stroke therapy, academic and industry group guidelines. And so I've outlined a few of them here, and these relate to both how preclinical testing is performed. So eliminating assessment bias and ensuring that you have statistical power are key things in doing these animal studies. And then relating to more the models, so testing this in models with complicating factors, evaluating how to best deliver the therapy. And so using the MotoTrack systems, as Drew talked about before, and I'm not going to get into any further, we're collecting a lot of data from a lot of animals. And so with these automated quantitative assessments, I'm going to go ahead and say that we can eliminate our assessment bias and we can ensure that we have sufficient statistical power. So now I wanted to talk about our use of testing our therapy now in some of these models with complicating factors. So today I'm going to focus only on advanced age on the interest of time. And so the reason is this is obviously a key consideration when thinking about developing stroke therapies, because advanced age it's the primary non-modifiable risk factor associated with stroke, and it's also associated with worse outcomes. With direct relevance to using VNS as a potential therapy for stroke, it's important to note that plasticity is widely believed to decline with advancing age. And so this is particularly important because if we think that VNS is enhancing plasticity to drive recovery, things like advanced age that limit plasticity may therefore also occlude the benefits of VNS. So we asked whether advanced age would interfere with VNS-dependent enhancement of recovery after stroke. And to do so, we conducted a trial that was very similar to the trial that I just explained, but we used 18-month-old Fisher rats. They were 18 months old when they entered the study. We see similar results, where prior to the injury, animals perform very well at the task, and after the injury, we see significant impairments. And these animals demonstrated a very modest recovery with extensive rehab over the course of 6 weeks. Once again, we see that VNS paired with rehab results in enhanced recovery at most time points during the training. And when we look at week 6, so after the cessation of stimulation, we see that these benefits are maintained and are resulting in significant improvements in recovery. So so to follow this up, I wanted to highlight another way you can use MotoTrack to look at the data. And so I've shown just sort of large-scale distilled down from group analysis, but here I wanted to get into a little bit more of the data. So what we're looking at here is a histogram of full peak forces at various time points of the groups during the study. And so we've binned the peak force on any particular trial, and you can see here for the aged animals we set the hit threshold at 100 grams. So you can see that primarily before lesion, most of the pull trials tend to fall in this above 100 gram, one of these bins that's above 100 grams, and very few fall below this threshold. So after the lesion, we see that both groups show a significant leftward shift where many more of these pulls are falling into these low peak force bins. And so this is consistent with a loss of forelimb strength. So these animals can no longer pull as hard, and we can see that borne out in these quantitative analyses here. If we look at animals on their 6th week, after their 6th week of rehabilitative training, you can see that there is now sort of a renormalization of this curve where there's a rightward shift. So now more of these pulls are exceeding the 100 gram threshold, but still many of them are down in the low pull force bins. If we look at animals who receive VNS paired with rehab, we see basically a renormalization or nearly a renormalization of pull forces compared to their pre-lesion performance. So this suggested to us that this is really a restoration of forelimb strength. So I don't want to oversell this and say that this is going to, they're 100% better, but it does look like on this measure at least that we are seeing a true improvement in performance, not some sort of epiphenomenon that could be based on the task. All right. So with all the caveats that go along with animal models, and especially animal models of advanced age, I'm going to go ahead and say that we have shown that at least, considering in our 18-month-old animal model, we see that VNS still significantly promotes recovery after stroke. So one of the other really important things when thinking about translating therapies is to determine the therapeutic window for which your therapy is effective. And so it's well known that most interventions become less effective with increasing time after stroke, and there's been some really nice work, molecular work that's been done to link this, at least in the context of rehabilitation, to this sort of of upregulation, so this push-pull balance between growth-promoting factors that tend to peak shortly after stroke and growth-inhibiting and plasticity-inhibiting factors that tend to peak later after stroke. So you sort of have this window during which rehabilitation tends to be the most effective. This is clinically important because there are many people, as I mentioned earlier, that are left with permanent neurological disabilities as a result of stroke, and it would be obviously extremely advantageous if your therapy would be effective in this population too, and not just people who are newly having strokes. So to test VNS's ability to see if we could deliver VNS later and it would still be effective, we performed a similar design of the study, but the difference here is that after we gave these animals a stroke, we allowed them to stay in their home cage for about 6 weeks, after which point we brought them back and assigned them to balanced groups to receive either rehab paired with VNS that I talked about before, or another interesting group here where these animals received rehabilitative training and they received a matched amount of VNS, but it was delivered 2 hours later each day. So show you what that looks like here. Again, our rehab and paired VNS groups look the same, but this delayed VNS group is going to get equivalent rehabilitative training, so 2 30-minute sessions each day, and then 2 hours after the last session of rehab, they're going to get a matched amount of stimuli. And so the idea here is that we can control for the sort of generalized effects with VNS. Since we think that VNS works by enhancing plasticity, we expect this temporal profile to be, we expect it to, it need to be temporally paired with neural activity related to the rehabilitation. So we hypothesized that delayed VNS would not be effective. So when we look at the data, once again, you guys are getting familiar with seeing these now. So animals in the rehab group are highly proficient at the task, and after the stroke they get much worse. And when we deliver rehab beginning 6 weeks after the stroke, you can see that they, once again, extensive rehab is beneficial, and these rats do demonstrate benefits as a result of the rehab. If we look at animals who get paired VNS, we see that they have significantly improved performance over the course of the therapy, and it extends at least 1 week after the cessation of VNS. And interestingly, if we look at our group who receives delayed VNS, we see recovery that's very comparable with animals who receive rehabilitative training alone. And once again, this suggests that VNS is not acting through some sort of generalized effect like some sort of anti-inflammatory property or an increase in neurogenesis, things that we wouldn't expect to require really precise timing. So this suggested to us once again that paired VNS is effective even when initiated many weeks after stroke. So I'm hard-pressed to say that we've defined the therapeutic window, but we've at least shown that VNS doesn't need to be delivered shortly after stroke in order to be effective, and it seems like if you deliver it, once again with all the caveats of animal models, that it might be effective when delivered 6 weeks later. So another thing that's obviously got clinical importance is evaluating the generalization of benefits. And so this is really important for therapies that are tailored towards enhancing rehabilitation. So you want to define the optimal way to deliver your rehabilitation. So you can imagine a scenario where patients receive VNS paired with a lot of different rehabilitative tasks and receive fewer stimulations, or you can imagine a scenario where a patient receives lots of stimulations on a smaller set of grouped rehabilitative tasks. And so to test the ability of the benefits of VNS to generalize, we performed a relatively similar study, but in this case, instead of using the isometric pull task, we used the supination task, where we trained animals on the task over the course of a couple weeks. We induced a cortical ischemic stroke. We allowed them to recover for 1 week, and then we brought them back and delivered VNS and rehab with the isometric pull task over the course of 6 weeks. At the end of that time, we switched them over to the isometric pull task, so they've never performed this task before. They weren't pre-trained on this task, and we simply assessed their performance on that task over the course of 4 weeks. And then we did sort of a long-term follow-up where we switched them back over to the supination task for an additional 2 weeks. And so what you'll see here is that on this supination task, you can see animals are very good at the task beforehand. They're significantly impaired as a result of the stroke, and they show very little recovery with extensive rehabilitative training. This task is very challenging for the rats to perform after the stroke. If we look at animals who receive paired VNS, we once again see the significant improvement in recovery during the time which VNS is delivered on the on the supination task. Now if we look, when we cross them over to the isometric pull task, what you can see here is that animals who receive VNS are demonstrating significantly improved performance on this similar forelimb assessment compared to rats who receive rehabilitative training alone. So this suggests that at least the benefits of VNS may generalize relatively similar forelimb tasks. If we bring them back and assess many weeks later, so on weeks 11 and 12, now we're assessing them once again on the supination task. You can see that we see very similar results to those that we saw about on week 6. And so I want to highlight that this is preliminary data, so we still have animals coming through sort of towards the end of the study. So this suggested to us, and once again, generalization is extremely hard to define because I obviously don't think VNS would generalize to improvements in hind limb function. It does show that the benefits of VNS will generalize to at least similar forelimb movements. And so the last thing we wanted to do was to ensure that this wasn't some sort of phenomenon that's restricted to our very specific way of inducing the stroke, was to see if we could use VNS to enhance recovery in other models of neurological disease. And so what I'm going to present, we've done this in a number of different models, but the one I wanted to present today was the one that's the most radically different from stroke. And so what we've done here is tested whether VNS can improve recovery in a model of peripheral nerve injury. And so peripheral nerve injury, many of you might be familiar with this, but the idea here is that cortical plasticity is associated with recovery after peripheral nerve injury, similar to the way it is associated with recovery after stroke. So we tested a variety of different nerve injury models, and we were able to do this because of this high throughput nature of these motor tasks. We tested a distal injury in the median nerve, proximal injury in both the median and ulnar nerve, one in which we just simply transected the nerve and then sutured them together, and one where we took a 5 millimeter gap out of the nerve and sutured the nerve ends into a tube. So once again, we trained the animals on the task, performed one of these nerve injuries. I'm sorry, for this study we ended up using the proximal gap injury. We performed that nerve injury. We allowed them in their home cage over the course of many weeks to allow the nerves to reinnervate the forelimb musculature. And then we brought them back and evaluated rehab without VNS and paired VNS, as I've shown you before. So if we look in this model, what you can see is that animals are extremely impaired, as you'd imagine, after a nerve transection. And they demonstrate modest but some modest but significant recovery with rehabilitative training over the course of many weeks of rehab. If we look at animals who receive paired VNS, once again we can see that VNS is enhancing the benefits of rehab and resulting in greater recovery in this model. So I didn't get a chance to show all of these today, but we've shown across a wide variety of mechanistically distinct models that VNS can enhance recovery in many of these models. So this gave us the confidence to go forward with the clinic. And so another reason VNS was really attractive, which I don't have too much time to give you a background on today, is that VNS is already FDA approved and it's safe and tolerable for patients. And so VNS is used a very different way to treat epilepsy. Basically, patients receive semi-continuous VNS rather than our very short bursts of VNS that are paired with specific events. But about 70,000 patients are already receiving VNS for epilepsy control and also for major depressive disorder. So our use of paired VNS to enhance plasticity uses about 1% of the total daily charge that's delivered with the FDA-approved levels of VNS. So this suggested to us that this might be even more well-tolerated than the semi-continuous use of VNS. And so when we translated this to a clinical study, we didn't get too creative. We basically used things that were very similar to how we conducted the rat studies. And the way the study was designed was that patients would receive an implantable vagus nerve stimulator and they would undergo rehabilitative training or physical rehab with a physical therapist. And they would perform various rehabilitative tasks, and when the therapist concluded that that was a successful task, they would press a button that would trigger this external device that would send a wireless signal to the IPG. IPG was implanted subclavicular in the patients, and the leads were placed on the vagus nerve. So when the therapist presses the button, we activate the vagus nerve much in the same way that we activate the vagus nerve during rehab in rats. And so this study was performed in conjunction with our collaborators at University of Glasgow. So this was spearheaded by Jesse Dawson, Dr. Jesse Dawson. And we recently published a study in stroke showing that in chronic stroke patients who received gold standard rehab alone, we saw a partial improvement in their upper extremity Fugelmeyer score. So the Fugelmeyer score is a relatively standard assessment of forelimb function. And patients who received VNS paired with rehabilitative training showed about a 3-fold increase in improvements on upper extremity Fugelmeyer function. And I want to caution that this was a very small study, and we're now moving forward in a randomized, double-blind, pivotal trial using this. And so this was exciting to us, and it really allowed us to do some of this two-way translation where we're learning things in the animal models and taking them to the clinic, and now we're learning things in the clinic, taking them back to the animal models. And these high throughput tasks have really been key to doing this. So with that, I wanted to wrap up and thank the many graduate students as well as postdocs and collaborators who have been really, who have guided much of this work, and specifically thank the undergraduate researchers who, this is where the rubber hits the road for getting stuff done with preclinical research. And of course, I wanted to thank my funding sources. So with that, I'm going to hand this back to Andy, and we're going to, I believe, open the floor up for the Q&A session. So I want to thank you for your time.
First question, this is for you, Drew. What system training is required and also provided by Vulintus to get labs up and going with the MotoTrack system?
Sure. So we have setup guides, of course, documents that you can use to kind of set it up by yourself. But in addition to that, we typically do video conferences when labs are first getting set up to discuss the setup, discuss training procedures, and really talk about how we can help train or help tailor the system to your particular use. And then we're pretty much always available for any kind of support, and we like doing that mostly through video conferences whenever possible.
Perfect. Very good. Okay. Stefano has asked, typically what is the duration for learning the pull task? Is this different between mice and rats?
Yeah, so for rats, it's typically about 3 to 4 weeks. It's a relatively quick task to train. For mice, it takes a little bit longer. It can often take about 2 weeks longer than that. A lot of that, I think, is due to somewhat fewer trials with mice with the labs that we've worked with. Mm-hmm. Labs that do food rewards tend to get a lot fewer trials, so you get fewer trials a day when you're training. If you do the liquid reward, you can get more. We don't really have too much data yet on how much quicker it is with the liquid training versus the food pellets, but I do believe the liquid reward helps train faster.
Perfect. Okay, and maybe part of this also a question about how many rats, mice, subjects could be trained simultaneously. Can you maybe touch on that, Drew, and then I'll ask Seth to chime in on what he's done in his lab also.
Sure. That's kind of a question of how diligent you want to be about putting rats or mice in and out of the systems. If you're running 2 30-minute sessions a day, as most of the labs we work with do, you can really take one animal out and put one rat in almost at the same time. It just takes a minute to basically stop one session and start the next. So if in a business day, you could theoretically run 8 animals in a day and still take a break for lunch. If you're not so dedicated, then you'd probably be talking about 5 to 6 animals per system. Most of the labs that we work with who want to do kind of larger studies and look at big groups of maybe 20 animals or 30 animals in a study, they tend to get multiple systems. I'm not trying to talk anyone into like, oh, you should buy multiple systems. But no, most labs that we work with, they start off with one system, they tend to like it, and then they scale up from there, usually up to about 4 or more systems.
Okay. And Seth, in regards to this question, kind of what happens in your lab on a daily basis?
Certainly. So typically the configuration is that we'll have a graduate student and then a graduate student or a postdoc or a technician working with 1 or 2 undergraduates to run one of these behavioral setups. And one behavioral setup typically for my lab consists of about 12 MotoTrack cages. And we run a couple of these. We have 4 racks of these things. So we're able to run many animals at once with a relatively small staff. So it scales pretty well.
Perfect. Very good. Okay, a question from Victoria. She has asked, can you share some effective examples of liquid rewards for mice?
Yeah, so April Becker used peanut oil. That was kind of a surprise to us in her methods paper, which you might have read. So she wanted to try and do the behavior without any kind of food deprivation or water deprivation. She's really big on trying to figure out very kind of naturalistic mouse behaviors. And kind of surprisingly, peanut oil was what she found. Where her mice did all that behavior in that paper without any kind of restriction. They did it just for the love of peanut oil. So that's maybe the first thing I'd suggest. Other than that, certainly doing any kind of liquid deprivation would give you a lot of motivation if you want to try that. I know that's like liquid deprivation is particularly dicey with animal models. Other than that, I would suggest sugary rewards. I don't have too much data on what type of rewards work best. I just have kind of the anecdotal stuff that I get back from the labs that we work with.
Okay. Seth, have you ever experimented with liquid reward or have any comment there?
No, unfortunately, I've only used rats, so I've used food pellets.
Okay, perfect. And actually, a good follow-up to this, there were questions from many in the audience, Chelsea and Lawrence. In your experience, does the researcher need to limit diet outside of reaching activities or the training stage, etc.? So, again, maybe Drew, you could provide the standard supported answer as Vulintus sees it, and Seth, maybe share what you've done in your lab.
Sure. So most labs we work with that do food rewards do complete food restriction during the week.
Okay.
And so the animals are not getting any supplemental food unless they're particularly slow learners. Because we're using an adaptive training algorithm, you can set the reward schedule so that they're still getting food even if they're not doing the task perfectly. And most rats or mice will get enough food to maintain their body weight just during training. So you don't really have to give them supplemental food. And then a lot of labs won't do work on the weekends because nobody likes to work on the weekends. And I found that if you give them free food on the weekends and you take it away on Sunday afternoon, it doesn't seem to hurt behavior. I think we've done a little bit of analysis and found that animals don't do as many trials on a Monday as they do on a Friday, but it's not a particularly dramatic number. I don't have the numbers off the top of my head, but it's relatively a small difference.
Okay. Seth, and how about from you?
Yeah, so Drew described how my lab does it, so I don't have a lot to add, but we monitor their weight closely, so they're completely food restricted during the week, full feed on the weekends, and we find that their food, especially with the adaptive algorithm, it's really stable. Their weights are, stay basically 100% of their weight when they go on food restriction.
Yeah, perfect. And I should add here too that in this case you want to make sure you're using a full diet food pellet that you can get from Bioserve or Purina. You don't want to be just using sugar pellets. You want to have something that has the full nutritional profile if you're going to try to get your whole food intake for the animal per day.
Perfect. Okay, that's great feedback. And actually, just going back to Victoria's question about examples of liquid reward, Donald in the audience has chimed in, and I believe he's suggesting something that he's used is diluted sweetened condensed milk. Apparently works well. So there you go, a suggestion from our audience. Thanks, Donald. All right, so next question, a question from Kristen. You mentioned someone is using the lever press test for bradykinesia. Do you have information on the output of motor impairment? And Seth, maybe you can take this one as I know that you've got some experience here.
Yeah, so my lab used this we've used this now in 3 models, I think, to look at bradykinesia. So we used it in a model of ischemic stroke, which I talked about extensively today. We used it in a model of hemorrhagic stroke. And we used it in something we haven't published in using some genetic rat models of Parkinson's disease. With the idea that hopefully we would be able to observe bradykinesia in those animals, but we never saw it. But in the first 2 models, we did see bradykinesia manifest in these animals as a reduction in or an increase in the amount of time, the interpress interval. So they're trained to press the lever rapidly within 500 milliseconds, and we see the interval grow longer after an injury. And so this is consistent with sort of a slowing down of forelimb function. So one thing we've never, it would be interesting to do this in some models that have other features of motor dysfunction. So for instance, things like spasticity. We expect that we might see an animal where they would be able to initiate a press and then would hold it and be unable to release it or press a second time. So there's some other things that you might be able to see related to bradykinesia in the lever test, but we've not followed that up extensively.
Okay. Well, that's a great answer. And actually, another one for you. Our audience has asked, Dr. Hays, can you share how VNS stimulation was delivered to the rats? And then also, sure, as a follow-up to this, actually, I'm combining 2 questions here. Can you also clarify the difference between VNS paired with rehab and then quote-unquote delayed VNS plus rehab? So that last example, I believe the last example that you shared, where there was a significant difference between the delayed VNS plus rehabs group. So just wait, I think the question is what that really means.
Certainly.
Okay.
So both great questions. I'm sorry I didn't talk about the delivery method for VNS earlier on. But the way this works is that animals receive just a standard bipolar stimulating cuff electrode. It gets implanted in the neck on the left cervical vagus nerve. We found that with left cervical vagus nerve, with these short bursts of stimulation, we don't see any changes in autonomic function, so especially cardiac function, which you can get if you stimulate the right vagus nerve.
Okay.
So the nerve projects bilaterally, so we get the same bang for our buck using left vagus nerve that we would get with either both or right vagus nerve stimulation. So it's an implanted cuff. Then the leads are tunneled up to a head cap, and then we plug the animals in with basically like a swivel cord connection that's plugged in during when they're to receive their VNS.
Perfect.
I'm my lab is certainly interested in talking about the parameters of VNS. But that's something, shoot me an email if you're more interested in that. I don't want to eat up, the rest of your day talking about that. So it's a great question.
A big discussion. Awesome. Okay. And maybe we can elaborate more on that too in the Q&A report when we pull that together following today's lecture. All right, a couple more questions. Again, this is a good Q&A, so I want to keep it going. Again, if the audience has to go, no problem. So a question from Chelsea Dunham: has any work been done to use this equipment as a physical therapy device for animals? And so she's referring here to treatment paradigms following like an orthopedic injury.
Right. So not to my knowledge. Mostly so far it's been used in either just neurological impairment models and we haven't really gotten into any kind of orthopedic models yet, but I'd be very excited to talk about that. Mm-hmm. It certainly is applicable. The tasks that the animals do after these impairment models is very similar to a lot of rehab tasks. I'm sure Seth could tell you a lot more about how those rehab tasks might be similar to various human tasks, but things like the supination task, turning a doorknob, that is one of the kind of key rehabilitation tasks that people do following a neurological injury. I'm not too familiar with what sort of rehabilitation tasks are done after an orthopedic injury, but our tasks should be pretty applicable to that.
Mm-hmm. Perfect. Okay. And a question for Maria. She just wants to know about your thoughts on using this system in light versus dark cycles, and whether that would affect the training process with the subjects and then the testing process. Again, do you know of any experience where people are looking at tests in either altered light dark cycles?
Sure. Yeah. So, I've done a lot of different types of behavior, not just motor behavior, and it certainly seems like testing during dark cycle tends to give you more activity. Mm-hmm. With the labs that we work with on the MotoTrack system, anyone who tests with mice tends to prefer dark cycle. They feel like they get a lot more activity out of that. With the rats, it's a little bit more variable. They seem to be a little bit more robust to being run in the light cycle when they might be in the dark cycle in their home cage. It's certainly not the case that animals just quit working during the light cycle when you might expect them to be asleep because they're nocturnal. So, they tend to fall into the routine that you set for them. But I do believe that running during the dark cycle for rodents is generally preferable if you can do it. It does mean you have to kind of bump your way around in the dark, but I do think there's a little bit of benefit for that.
Okay. Seth, anything to add on this subject? Has any of your research ever included these type of considerations?
Yeah, I mean, we've considered it, but we've just sort of based on what Drew said, we've not seen big changes. We test them during the light cycle, and we see a couple hundred trials a session. So mm-hmm. It's not been a big. It's not caused us to change anything, but it's certainly a consideration, and it's actually a really interesting question. It's an experimental question.
Perfect. Well, that's great feedback, guys.